Cool. I can see it. You're good to go.
And I got the right view up there too. So excellent.
Thank you.
All right. Well, thank you for that introduction, and thank you all for the invitation to come here and chat with y'all today. And so I'm going to jump right into it. What I'm going to be talking about today is, like Adam said, a comparison of methods, comparing some eDNA methods and another really exciting approach, which is scent detection canines for the detection of zebra mussels. What I'd first like to do is do a quick sort of shameless plug as an introduction here, and talk about this special issue, or they call them research topics, in Frontiers magazine that I was involved with, and got to co-edit along with Katie Klimas, who I saw is on this call, and our colleague Hiroki Yamanaka, who's doing really great eDNA work out of Ryokoku University in Japan. And we edited this issue that we called Environmental DNA Innovations for Conservation. And I just wanted to point it out that it's available online, and it contains some really nice studies that pushed eDNA, or that we were trying to solicit studies that were pushing the boundary of what eDNA can do. And so as we were editing this volume, we noticed that there were a couple of distinct trends that emerged across the submissions we received, and that has helped us realize that innovation in eDNA application comes in many different forms, and whether it's too corny or not, I think the jury's still out. It probably is too corny, but we envision these as four key bases of innovation in the field of eDNA application. And so the first base of innovation that we saw had to do with the foundation, or we called it foundation innovations, and they describe the advancement of our understanding of aspects of really foundational ecological aspects of eDNA, right? Even though it's been, even though I think this field is occasionally overlooked during the excitement over the last decade of sort of what can we detect now, and where can we detect it. The ecology of eDNA really represents this ecological, or eDNA itself represents this ecological entity that's worthy of study in its own right. Perhaps the most consistent form of innovation in eDNA applications has been that excitement of confronting eDNA methods with new species, and in new types of ecosystems. And there's been a lot of great work recently looking at even moving from the water and the land, and now looking at airborne eDNA methods and being able to detect species, plants and animals, using dust and air samples. And so these we call these application innovations for eDNA. We also, in that research topic, or that special issue, saw a lot of what we call calibration innovations, and these are incredibly useful comparisons between eDNA and more traditional survey methods, and they've been useful to contribute to the quantification of both the sensitivity and the accuracy of what we do with eDNA analysis. And I would argue that they represent a really critical link between eDNA applications and the existing toolboxes for eDNA for conservation and ecology study. And so the continued growth of calibration application innovations is really important. And then finally, we refer to innovations that advance the potential knowledge gained from eDNA analysis as interpretation innovations. So these previous three types of innovations all provide the foundation for research, which pushes the bounds of what we can learn using eDNA analysis. So here today, while I encourage you to check out that research topic at the Frontiers website. It's got 18 awesome, innovative papers, including several from several of y'all here in the room. But with my time here today, I'm going to talk about a recent project from my team and some collaborators that calibrated our eDNA work against another novel sampling method. And our target is going to be the zebra mussel, Dreissena polymorpha. And I won't belabor the introduction too much here, except to say that these are Eurasian mussels that were first detected in North America in the 1980s in the Great Lakes, almost certainly due to being introduced as hitchhikers on ballast water, or in ballast water, I should say. They grow these amazing colonies over any hard surfaces, as shown by this iconic image of a discarded grocery cart. And I think this cart is useful because it gives us a nice nod to the economic impact that these creatures have. It costs millions or even billions of dollars annually to clean power plants, municipal water intakes, and really all the other hard structures that these mussels foul. Hard structures also include uniotid mussels and other hard native critters, and so the introduction of zebra mussels is a biodiversity concern as well. And then finally, the zebra mussels are a voracious filter feeder, and so they can dramatically alter plankton and energy flows in aquatic food webs. And worse yet, they have this ability to selectively graze and avoid the bad stuff, the harmful algal species, while eating the good algae. And by eliminating that competition to the harmful algae or the cyanobacteria, they can actually contribute to algal blooms that affect wildlife and human water use. Zebra mussels in Texas first appeared in Lake Texoma on the U.S. or the Texas-Oklahoma border back in 2009, and over the past decade or so, they've spread rapidly throughout the state. I'll also give a quick mention here that last year we had our first detection of quagga mussels in the state in Lake Amistad on the Texas-Mexico border. And although my talk here today is going to focus on zebra mussels, we're in the process of reevaluating a lot of the samples that we previously collected and archived to see if maybe quagga mussels were present perhaps earlier than we had originally thought. The appearance of zebra mussels in Texas has led to a management response that focuses on prevention, like the education and awareness campaign you see here, Protect the Lakes You Love, Clean, Drain, Dry your boats. And today I'm going to talk about, prevention and early detection and rapid response approaches, including canine scent detection and the analysis of eDNA as two tools in the early detection and rapid response wheelhouse. I'll point out that while like you can see in this picture on the top here, while canines have been used successfully to inspect watercraft and look for adult mussels, what I'm going to focus on in this talk is about how these methods can be used to detect zebra mussel essence in environmental samples, right, including their eDNA, and things like their larval veliger, free-swimming veliger larval stage, and other cells, mucus, debris that these filter-feeding adults might shed into the water. And so we're going to look at we'll briefly look at three big questions here today. First, just a practical question. Can we train canines to detect this zebra mussel essence, right? Not the adult mussels, but instead the small stuff, the stuff you can't see. Number two, what are the limits of detections of both canines and eDNA for zebra mussels? And then I'm going to finish with a quick evaluation of some of the strengths and weaknesses of the two approaches that we observed. And so we'll start with the dogs. And of course, we have to start with a picture of the doggos themselves. So today I'm going to present the results, the data from 6 canine collaborators. Each of these dogs is an employee of a group called Muscle Dogs, based out of California. And so their main gig, right, the way they pay the bills is doing watercraft inspection at boat launches, looking at boats entering and leaving the water, and looking for those adult mussels. But again, here we're looking to see if they could detect the smaller stuff, which we could practically consider analogous to eDNA, and in those environmental samples. And so the canine training uses this thing called an olfactometer. And briefly, what I've got a little video here. I'm going to explain it really quick. The video is probably more useful than the engineering schematic here. So what the olfactometer is it's this machine that has the capacity to hold vials that contain different odor sources. And then there's this laptop computer that's hooked up to the olfactometer that can automatically and even randomly assign airflow from those odor sources out the front of each of 3 ports on the front of the machine. And the port also has this infrared beam in it, so that it can measure how long the dog sticks its nose in these different ports, and in other words, how strongly they're reacting to the odor that's being put out. And so I'm going to show you this video here in just a second, and we'll see if the sound's set up correctly. But hopefully what you'll hear is, in this training exercise, the computer is assigning a zebra mussel scent to one of 3 ports, and then the other 2 ports are receiving no odor cue. And you'll hear an alert, a beep, like a whoop, when the dog places its nose in the correct zebra mussel scented port for a long enough time. And then what you won't see in here, but for the purposes of training, we can also have the machine give kind of a buzz noise if the dog signals the wrong port. This can be done either blind or such that the trainer knows whether the dog's picked the right or incorrect port. And so you're going to see a training exercise here where the trainer hears that the dog gets it right, so we can reinforce that with a treat. But then in some of the later trials I'll talk about here today, we can turn those signals off, and we can turn even the awareness off. So even the trainer, the handler, doesn't know, is blind to which, if any, of the ports contained a zebra mussel scent. And so let me play this video here. So that's the olfactometer. I don't know if you could hear that or not, but the dog held her nose in the right port for long enough in that middle port. She heard that beep. She knew she did a good job. She's a good dog, and she turned around excitedly for that treat. And so that's the method that we can use to train these dogs to, there we go, to recognize the zebra mussel scent essence. And so to get to one of our major punchlines really quickly, we figured out early that the dogs couldn't just detect zebra mussel in a surface water grab. So all the sample preparation today will begin by having to generate this concentrated plankton sample through multiple tosses of a plankton net, an 80 micron mesh vertical plankton tow, and we would fill up a vial, a 2-liter bottle with repeated tows, and then we would sieve that and concentrate those samples even more, use microscopy to quantify the zebra mussel veligers in particular in these samples. This is also a really good point just to really quickly point out, especially for the managers in the room, that throughout everything I'll be talking about today, we did use really exhaustive decontamination procedures to prevent the spread of zebra mussels. And so in particular for our nets and other things that contacted the water, we soaked them in vinegar for an hour, followed by a 10% bleach, soak for another 10 minutes, and then copious rinsing with water and complete drying overnight, so that so that we didn't spread anything. And we had the relevant Texas Parks and Wildlife exotic species research permits, and approval from our institutional animal care and use committee for working with our dog co-stars here. Okay, so now back to the first question. Can we train dogs to detect zebra mussel odors in natural waters? We worked in a series of so all of this work today was conducted in a series of zebra mussel positive and negative lakes near San Antonio, Texas. And for these first trials, we concentrated plankton, including zebra mussel veligers, from Canyon Lake, north of San Antonio. And then we resuspended that material either in spring water to start, or from water from a zebra mussel negative site. And so the idea here is that we started really simple and then made our environment increasingly complex as we assessed the dog's ability to discern. So minimizing as many background odors as possible, we prepared a 5 milliliter sample, about 900 veligers per milliliter in spring water, and examined whether the dogs could differentiate between that and a 5 ml sample of spring water only. And so here we see the proportion of times that the dog got that trial, that choice, which is the 1 zebra mussel port out of the 3. We see the proportion correct, and it was for most of our dogs close to either 8 out of 10 or 9 out of 10 trials, or 10 out of 10 trials, that they could discern the zebra mussel port from the other 2. Now, the next and critical part of the method that I learned a lot about from my animal science colleagues was that we had to evaluate performance when we gave the dogs all clean water, right? In other words, no positive sample, no zebra mussel positive choice. And the reason this is important is because the dogs are really sneaky and really clever, and they'll use any possible advantage to make the right choice to get that treat. And so if there's some kind of auditory, like a little click or a different vibration or something that maybe we haven't even thought of or can't even detect, but that it helps the dogs to figure out what the right choice is, then they could actually identify the correct port. I should say correct port. The machine can give 3, each port would contain no scent, but it would still assign one of those ports as the winner, right, where the dog should spend most of its time. And the dogs could identify that even if no odor cue was provided. And so if the dogs can't cheat, then we would expect them to get the right port, 1 out of 3, have a 1 out of 3 chance of getting it. And that really is what we saw. So here, for a negative control, right, no odor presented, our dogs ended up getting the right answer. It turned into a game of chance, right, 1 out of 3. Once the dogs got really good at discerning, the zebra mussel water or zebra mussels in spring water, then we trained them with more complex samples, filtered lake water from a negative site with the zebra mussel plankton sample diluted in that. And again, our dogs performed really well. Okay, so one limitation of what I've shown you so far, though, is that during that Veliger concentration step, there was a lot of, we didn't concentrate just the zebra mussel Veligers, although they certainly were a considerable proportion of the Veliger, of the plankton samples. We may have been inadvertently training our dogs to detect not just Veligers, but rather that increased plankton load, that increased biomass in target samples compared to the non-target, just due to the concentration step. So to confirm that it's the zebra mussel signal that the canines were responding to, we conducted a test in which plankton was identically concentrated from several lakes and reconstituted into filtered lake water. And we did this. We also added another positive zebra mussel site, Lake Placid in Texas. And good news here is that the dogs continue to demonstrate a high ability to discern those zebra mussel samples from the non-zebra mussel samples, even in a novel plankton and water combination. Okay, so on to the exciting part. Let's introduce some eDNA into this as well. We want to know what are the limitations of detection of both the canines and eDNA analysis. And so we're going to follow similar methods here, except that we created a dilution series in half log steps, and we made 3 identical sets of serial dilutions for canine testing, and then we set up a 4th set of a serial dilution that we exposed to eDNA detection. The eDNA methods for this group, we filtered using a 1 micron polycarbonate filter. We extracted the DNA from that filter using a CTAB chloroform extraction. We used a TACMAN-based assay, hydrolysis probe-based assay that targets zebra mussels site B specifically. And I'll show you some data on our limit of detection, which we defined as 1 of 3 of our technical replicates in our dilution series being positive, and our limit of quantification, which I'll define as all 3 of 3 technical replicates being positive. And so the rate of eDNA positives within sample replicates ranged from, across our series, we got anywhere between 0 of 3 or 1 of 3, all the way up to 3 of 3 replicates, technical replicates being positive. And based on the amplification that we saw, the limit of detection was less than a Veliger per milliliter, down to 0.02 of a Veliger per milliliter. And the limit of quantification was around 26 Veligers per milliliter, which keep that number in mind in particular, 26 Veligers per milliliter. Let me show you how we tested the dogs. Again, we used the olfactometer, and we used this this method called a 2 down, 1 up descending staircase procedure. And so in this procedure, if the machine now is doing this blind, the computer can blindly increase or decrease the concentration vial that it presents to the dog as the positive signal. And so when the canines make 2 consecutive correct responses on a concentration, the computer says, okay, the dog understands that concentration. I'm going to decrease the concentration by 1 step or a half log concentration. When the dog makes an incorrect response, the computer moves the dilution back up 1/2 step, and the testing continues until the dog reverses directions 8 times or 40 trials commence, and then the dog's just getting tired by that point. And so here we see we've got the concentration, the odor concentration on the left. This is just an index, not the actual concentration. And then on the x-axis is the number of trials. And so you see here, Dog got 2 trials correct, and then so the computer said, all right, let's make it less concentrated. Dog gets it correct again. They try it again. The dog gets it incorrect, so it goes back up to a less concentrated, or to a more concentrated level. And every time the dog gets 2 detections correct, we see it goes down a step, until eventually we've reached that threshold where the dog seems to be getting just as many correct as they are incorrect. And here we're looking at, broken down by dog, the probability of a positive detection related to dilution factor. So the vertical lines on each one of these graphs shows that each individual dog's 75% accuracy detection threshold, where they get 3 out of 4 samples correct. That horizontal line gives you a 95% confidence interval around that. And so some take-home messages here are that 3 of our 6 dogs showed detection limits within a 15 to 38% dilution of that original sample. And lower limits ranged from anywhere from 2% to maybe 0.1% dilution of the original Veliger sample, suggesting, overall that our canines could detect a limit somewhere between just about 1 Veliger per milliliter, and some of our worst dogs. Captain is a good dog, but not necessarily good at this particular exercise. And so his limit of detection is somewhere on the order of a couple hundred Veligers. Let me throw up the eDNA limits of quantification, which were really similar to, it turns out the limit of quantification was really similar to canines, with all replicates quantifying a concentration. The eDNA quantification limit was about 26 Veligers per milliliter. This exceeds the 75% detection limit for all but 2 of our canines, and exceeds the 100% detection range for all of our canines. And so overall, we can say that the eDNA appeared to be 1 to 100-fold greater sensitivity in terms of quantification, or what we would call quantification, right? That 75% detection, and then the limit of detection is something like 30 to 12,000 times greater for eDNA than it is based on which dog we were looking at. Okay, so finishing up, let me talk really quickly about some of the strengths and weaknesses of these 2 approaches, and we're going to throw microscopy in there as well. And so to guide this discussion, we confronted microscopy, canine detection, and eDNA analysis with completely blind I went out and collected them, but to the graduate students, the poor graduate students that had to do the microscopy and the eDNA methods, or to the dogs, these samples were blind. And just jumping right into the punchline here for the sake of time, using microscopy, okay, and so what we've got here, we went to 10 different lakes. Here is how Texas Parks and Wildlife, in this first column, how they categorize the lake. So 7 out of 10 Texas Parks and Wildlife classifies as positive for zebra mussels in some form. They've got kind of a tiered system of how positive a lake is that I won't get into much detail. I won't get into detail about here. And then we sampled 3 that Texas Parks and Wildlife thinks are negative for the presence of zebra mussels. 5 out of the 10 sites is what we found using microscopy to contain Veligers. And so we missed a couple of sites. And my graduate students wanted me to point out that overall, this method is time consuming and physically straining, and apparently has the least accuracy as well. And so if dogs or eDNA can offer any kind of improvement on that, there's multiple benefits. And when we look at the dog and the eDNA results, I think the main story here is that we saw a lot of consistency, except for a couple of cases where we didn't. Both dogs and eDNA identified really low levels of zebra mussels in Lake Allen Henry, which is a lake that Texas Parks and Wildlife and our microscopy suggests that zebra mussels do not occur. So we're working with the agency to learn more about that particular site. Texas Parks and Wildlife classifies Granger Lake as having some level of zebra mussel infestation, and we failed to detect zebra mussels with any of our methods. Dogs and eDNA detected zebra mussels in Lake Georgetown, which Texas Parks and Wildlife calls positive, and we failed to detect using microscopy. So perhaps the grad students' eyes were strained at this point. And then the dogs were the only group to categorize Choke Canyon as a positive site. And I'll point out that Nate Hall, the leader of the PI from our canine olfaction lab, is particularly nervous about this result. And so we're exploring that further and trying to understand why the dogs might have detected there, given that all the other evidence suggests that there's not zebra mussels present. And so together, these results indicate that the canines show, it's really relevant to realize that our canines had a lot of individual variability between one canine to the next in terms of sensitivity. The least detective canines showed decreased performance starting at several hundred veligers per milliliter, but we had another canine that had detections that approached 1 veliger per milliliter. And so, interestingly, this suggests that the odor signature of zebra mussels is still available even when the veligers themselves, the organisms themselves, are really rare. And that our dogs are really well-trained and really sensitive dogs may be approaching what we might call eDNA kinds of detection. Importantly, the dogs give you that real-time advantage. So they may be a little more error-prone, but they can tell you on the site whether zebra mussels are present. Saying whether they're present or not may be a little too bold. eDNA is a little bit slower, but appears to be less error-prone and more sensitive. And avoids the need for any kind of sample concentration. So I feel like I flew through that, but I'm going to stop here. Thank y'all again for the invitation to talk to you, and hopefully we can give you a chance to ask some questions. If we don't get to everything, my contact information is here, and I would be happy to continue to chat with you even after the meeting here today.